jazz. Oh, that's right. I have to click the share button. I completely forgot. Okay. Oh, please, dear God, let my computer not crash. Okay. Well, it's good to see everyone who's here. Welcome back, TriBeta members, as always. So I've already said this enough this semester, just the general housekeeping stuff, 12 hours to be considered an active member, attend 6 out of the 7 meetings. You can also email any one of the officers in case something comes up, like, hey, I got sick, or an accident happened, so I couldn't attend this meeting, and that's a-okay with us, just as long as you communicate. And as always, officer elections and new member inductions will be held in the spring, and that will be on not the 22nd, but 2 weeks before the 22nd, because for the 22nd is our trivia night. So as always, trivia night will have multiple different questions ranging from different categories. However, it will be held over Zoom on Kahoot. That way we can all social distance, be safe from each other. And you're also free to disperse the link how you see fit. So we'll send it out to the biology faculty. We'll also send it out to whichever organizations would like to participate. So anyway, just be a fun free event, just a nice little stress reliever before finals and all that jazz. All right, and here we have some ideas for TriBeta gaining hours, specifically with TriBeta events. Obviously, you have our meetings, trivia night, tutoring, and the Southern Central, regional convention, April 10th. Again, it is a $25 registration fee, and it will be held on April 5th. And then, as always, the volunteering that we've had. And we will be having a state park cleanup on the, I just said this, it's the 20th, right, Morgan? Okay, yeah. So we will be having that on the 20th. And basically we'll just be broken into groups of ranging from 10 people and no more than 10, if supposing more people do come than 10. And we'll just be cleaning up trails and getting things ready before Easter. So that's what the park ranger pretty much had for us. All right. If you miss a meeting, as always, we record these on YouTube, and if our professors are more inclined to allow our members to see it on YouTube and post it, then we will upload it to YouTube, and our members will be able to check it out at any time of their choosing. All right. And today's speaker is Dr. Galliard. Oh, good Lord. I'm sorry. And she will be speaking with us on her research. So I will stop sharing my screen, and it's up to you.
Okay, so let me just share my screen. Everyone can see that? Okay.
Yes, ma'am.
Let me just move my window. Okay, well, I'm going to tell you a little bit about the research that goes on in my lab. And I'm sorry, I'm trying to move my window. There we go. So the organism that my research lab works with is Chlamydomonas reinhardtii. And this is a unicellular algae found in many different environments around the world. It has 2 flagella, which we actually now call cilia, and it uses those for motility. It is a haploid organism, so that makes it very convenient for a lot of genetic studies because you only have to worry about 1 copy of the gene typically. And what's also really great about this organism is that the interior structure of the cilia, which is called the axoneme, which is like a nano machine, it can be biochemically isolated completely away from the rest of the cell. And that's not a trivial feature because in a lot of other organisms that have cilia, it's not that easy to do. And so here you can see some images of Chlamydomonas. And so I wanted to talk for just a moment about how ciliary motility is regulated. So again, in the upper left here, you have Chlamydomonas cell. And so it's got its 2 cilia. But in the human body, there are many, many different locations for cilia. So for example, what you see here on the right is the airway. So this is a human epithelial airway. And you can see that there, excuse me, many, many cilia on this surface. So regardless of whether you're talking about a unicellular organism like Chlamydomonas or an airway of humans, the cilia and their interior structure, the axoneme, is highly conserved. So on the inside, you have the axoneme, which is what we call a 9+2 structure. So it's got 9 doublet microtubules around the periphery and then a central pair of microtubules. And so what you're looking at here is a cross section through a cilium showing that 9+2 axoneme composition. And so again, it doesn't matter what kind of eukaryotic organism you're dealing with, the structure of the cilium is highly conserved. And so that makes Chlamydomonas an excellent model for studying human cilia because of that structural conservation. And I do want to just mention cilia are unique to eukaryotes. So when we think of bacteria and bacterial flagella, those are structurally and evolutionarily completely different than what we have in eukaryotes. And in fact, that's why we're trying to exclusively use the name cilia now, to distinguish between eukaryotic structures and the bacterial flagella. So this is just more information about the axoneme. And you can see that, again, those 9 outer doublet microtubules and the central pair of microtubules. The take-home message here is that there is a lot of intricate structure to the axoneme, and all of these structures are important in regulation of motility. And again, this is highly conserved among different species. So what drives the movement of the axoneme and hence cilia is a motor protein known as dynein. So dynein is sorry about that. Dynein is a motor protein that hydrolyzes ATP for energy. And there are dynein motor proteins shown here in red throughout the axoneme. And those are all very important to create the bending that is characteristic of ciliary motility. So what's cool about the axoneme is that it is a biological nanomachine in which everything that's needed for motility is built into that structure with the exception of ATP. But if you were to add back ATP artificially, you can make dead cilia move. And so it's a really, really interesting system in which you can manipulate the conditions and then test how those conditions affect motility. So again, dynein is the motor protein that drives the movement. And the way this works is that dynein walks along a doublet microtubule, but it carries the adjacent or neighboring doublet microtubule as a cargo. And so if they weren't all connected in the axoneme, when dynein becomes active in the presence of ATP, one doublet microtubule will slide right past the other. However, because they are confined in that axoneme and linked together with linking proteins, the doublet microtubules instead bend. And that is really the force that generates movement. So I mentioned previously that axonemes can be isolated, and that's what makes it a really good model organism. So what we do is we deflagellate, as we call it. So we remove the cilia from the cell body, and then we demembranate those cilia. So we use a detergent in order to strip away the membrane and other soluble components of the cytosol. And what you're left with is that nanomachine, the axoneme. So there have been a lot of genetic studies in Chlamydomonas, and these studies have revealed many different genes that are important in regulating motility. And for many of those genes, they code for a structural component of the axoneme. So this is a subset here, and you can see that there are mutants in the outer dining arms, the inner dining arms, as well as the radial spokes and the central pair, just to name a few. And so what's very interesting about this as well is that many of these genes have homologs in humans, such that when human homologs of these genes are defective, it causes problems with ciliary motility. So why would we be interested in understanding ciliary motility? Well, that's because there are a lot of what we call ciliopathies in humans, and I'll talk about those in just a moment. But one of the questions that we're asking is we're taking a look at a new mutant. Okay, so there's a lot of new mutants available that we didn't have before. And we're taking a look at a mutant that is defective in what's known as radial spoke protein 1. And radial spoke protein 1 is part of this radial spoke head that you see here. We know that the radial spoke head is important for regulating motility, but we don't completely understand the function of radial spoke protein 1. And so one of my new graduate students is investigating a mutant of Chlamydomonas that's defective in this protein. And so he's going to take what's known as a reverse genetics approach, where you interfere with the function of the gene, and then you determine what phenotype results. And so that should tell us more about what this protein does. Incidentally, there are many examples of humans with defects in the homolog for radial spoke protein 1. And when those humans have defects in this protein, their cilia do not function properly. And so why do we care about this? Well, when cilia don't function properly, it can cause what's known as primary ciliary dyskinesia or immotile cilia syndrome. And this is characterized by respiratory tract infections, which can often result in respiratory failure, as well as sinus and middle ear infections, sterility in males, and reduced fertility in females. And so the first two bullet points are fairly easy to understand because our respiratory tract and our sinus cavities in our middle ear all have motile cilia that help to either sweep particles out or keep fluid moving. And so when these cilia don't move, it causes disease. Sterility in males, well, they're immotile sperm. And so if sperm don't swim, then that decreases fertility. And for females, there are motile cilia in the fallopian tubes that help to sweep the fertilized egg down into the uterus for implantation. And when these cilia don't move, then that can cause a tubal pregnancy, which can be very dangerous, and overall reduced fertility. So these are some of the reasons we are interested in understanding more about what goes wrong with these cilia. Another very tragic effect of immotile cilia is something called hydrocephalus. And so there are cavities in the brain that are filled with fluid, and these cavities are connected with the interior of the spinal cord. And so this cerebrospinal fluid needs to stay circulating, and motile cilia are the ones to keep that fluid circulating. If the cilia are immotile, then the fluid can build up in the cavities of the brain and cause what's known as hydrocephalus. And hydrocephalus is literally water on the brain. And you can see this poor infant here exhibiting hydrocephalus, and they've placed a shunt in the baby's brain to drain off that excessive fluid. In addition, immotile cilia can cause what's known as situs inversus. And when situs inversus is caused by immotile cilia, it's called Cartaginer syndrome. And essentially what happens in Cartagener syndrome is very early in embryonic development, there are motile cilia that are essential for sweeping a chemical from one side of the embryo to the other. And that gradient of chemical that is established sets up the left-right axis of the developing embryo. So if there's no motile cilia, then there's no left-right axis established, and instead organ placement is randomized. So for example, you see in this chest x-ray, the heart is on the right side of this person's body, and it should be on the left. And so about, I would say 20 to 30% of the time, the heart being on the wrong side causes problems with the vasculature, because the vasculature is designed, you know, to connect to a heart on the left side. When it's moved to the right, sometimes that the whole vasculature doesn't form properly, and it causes essentially what's known as a congenital heart defect. So with respect to understanding ciliary motility, another project in my laboratory is taking a look at the chemicals of electronic nicotine delivery systems, or vaping systems, and determining how they might affect ciliary motility. So there are a number of different chemicals in vape. You know, there's a lot, more than you might think. And we don't really know the effects of these chemicals on the lungs, on long-term health, and certainly not on ciliary motility. And so a project in my lab, it's currently being done by Jessica Ridland, is to test the different components of the vape chemicals and determine if they have an effect on ciliary motility. And so I didn't have time to put any data from that project in this presentation because it's relatively new project, but suffice it to say that indeed some of the chemicals already we've seen arrest ciliary motility. So why might that be a big deal in a developed human? Well, you know, if you continue to vape and that causes your cilia to be immotile, it can make you more susceptible to respiratory infection and asthma, for example. So people should know what they're getting into when they vape, and those studies just haven't been done. So she's working on that. Now, another aspect of my laboratory that's totally different from studying ciliary motility is taking a look at programmed cell death. And so programmed cell death is a genetically programmed, controlled, neat kind of cell death. And believe it or not, unicellular organisms exhibit programmed cell death too, including Chlamydia monas. You can see these Chlamydia monas cells here. If you were familiar with Chlamydia monas, you would know that these look kind of bumpy in the image, and the cilia are gone. So these are cells that are undergoing programmed cell death. So again, programmed cell death is genetically controlled. It's considered an active and regulated cell death. And there's a specific type of programmed cell death in animal cells that is known as apoptosis. And so you might be more familiar with that term, but it's a type of programmed cell death. And one of the keys to apoptosis is that there are enzymes known as caspases, which regulate that apoptosis. Caspases are proteases that cleave proteins, and so they are very important in the apoptosis pathway. Well, in general, programmed cell death is distinct from another kind of cell death known as necrosis. And necrosis is a very accidental, acute, messy cell death. For example, if there's a sudden injury, then cells might die by necrosis. So there's nothing planned or programmed about that. It's just, you know, a mechanical injury to the cell, for example. On the left side, this is a cell undergoing programmed cell death, and you can see that it is breaking apart on the inside. So its interior components are breaking apart into bits, but notice that the plasma membrane stays intact. On the far right, in contrast, is a cell undergoing necrosis. And so you can see that the intracellular contents are spilling out into the external environment. So the plasma membrane is not intact, and this actually can cause an inflammatory response. So when you have a mechanical injury, one of the reasons you may get inflammation is because you have the intracellular content spilling out into the extracellular environment. And so the immune system recognizes those as not normal to be there in that context and mounts an inflammatory response. In multicellular organisms, cells undergoing programmed cell death will often be engulfed phagocytically by cells of the immune system. So they will actually eat the dying cells and clear them from the body. In eukaryotes, these are some features of programmed cell death. So you have cell shrinkage, you have proteolysis, again, proteins being chopped up. You also have what's known as DNA laddering, and that's when DNA is chopped up into distinctly sized fragments, such that when you perform DNA electrophoresis, you see bands of distinct sizes. It kind of looks like a ladder, which is why it's called laddering. Vacuolization describes the organelles breaking apart into little bits. The cytoskeleton will also fragment. In addition, reactive oxygen species will be released. And lastly, another key feature is the phospholipid phosphatidylserine, which is a component of the plasma membrane that's normally found on the internal side of the plasma membrane's lipid bilayer. It actually gets flipped to the outside of the plasma membrane's lipid bilayer. And that's a signal that the cell is dying, and that can serve to signal to those phagocytic cells to engulf that dying cell. Well, as far as animals go, programmed cell death is very important during development. So for example, programmed cell death is essential for digit sculpting. So the fact that fingers and toes form is because cells in between our digits undergo programmed cell death at a certain point during embryonic development. So here in A, we see a mouse limb, and you can see that in bright green, there are cells in between the future digits. All of those bright green cells are undergoing programmed cell death. And once programmed cell death has occurred, you can see the results. You have digit sculpting. For example, another classic programmed cell death event is during metamorphosis of the frog. So in transitioning from a tadpole to an adult frog, the cells of the tail undergo programmed cell death and die, resulting in the adult frog body. And one other that's maybe a little more relevant to us as adults is that programmed cell death is important for removing misbehaving cells in our body. So for example, precancerous cells are often recognized as such and are programmed to die. And so when those precancerous cells begin to undergo programmed cell death, again, a type of that being apoptosis, the cells break apart into apoptotic bodies. And again, cells of the immune system often engulf the cells and the cell bits in order to clear them from the body. So programmed cell death protects us against diseases like cancer. So what's really weird is, why would a unicellular organism undergo programmed cell death? I think we can understand why a multicellular organism might do it, right? If you destroy one cell to the benefit of the rest of the organism, well, it's easy to understand why that would have evolved. But in a unicellular organism, it doesn't really make sense. So if it's just one cell, why would that cell destroy itself? Because that's the end of the cell, right? That there's no organismal benefit to that. It's a suicide in a sense. And so one of the hypotheses about this is that could cells like Chlamydomonas be exhibiting some kind of altruistic behavior? So will one cell sacrifice itself for other cells? Well, altruism is very controversial in this sense because it's not really thought to be possible that it could have evolved. Meaning that if something is truly altruistic, then what is the gene that would have selected for that? For example, what would be the benefit? How would it ever get selected for? Well, maybe what we're seeing is not truly altruistic. So if we look at a population of Chlamydomonas cells and we see that some are undergoing programmed cell death, it may not be that it's truly altruistic and one cell is destroying itself out of complete sacrifice for another cell. Because many Chlamydomonas cells live in colonies. And in these colonies, the members of the colony can be clones of one another. So Chlamydomonas is capable of asexual reproduction. It can also reproduce sexually, but asexual is sort of the everyday mode. And so you have this population of Chlamydomonas cells that's all genetically identical. And so if one cell dies, it actually would benefit its clones or direct kin. And so that's kin selection, which is not truly altruistic, right? So even if we die, right, we may have relatives that share our genes that will carry those genes on to the next generation. So I don't have any children of my own, right? So evolutionarily, my fitness maybe isn't as good as people that do, but I have a niece and a nephew, for example, right? And so we share a quarter of our genes on average. And so my fitness is enhanced if I take care of my niece and nephew because they're going to pass on those genes, right? And so this is kind of the same idea. Perhaps organisms that live as clones have evolved programmed cell death as a way to benefit their clones, not because it's truly altruistic. Does that make sense? Okay. So how might the death of a cell benefit its clones? Well, for example, if that cell who dies is infected with a pathogen, well, that may annihilate the infection for the whole population. And again, because the other members of the population are clones, then that benefits the dying organism genetically in a great way. Also, what if the dying cell has damaged DNA? Well, its self-destruction could improve the gene pool of the population by preventing that damaged DNA from being passed on to the next generation. One of my favorite hypotheses, and there's actually some evidence for this, is that a dying cell will make nutrients available for the surviving members. So it releases contents that can be utilized as nutrients for the surviving members. And in general, just may aid in reproduction of the organism as a whole. So I mentioned that Chlamydomonas exhibits programmed cell death. Well, many other unicellular eukaryotes do as well, including yeast. So this is Saccharomyces cerevisiae, which is the brewer's yeast or budding yeast or baker's yeast, all the same. And it also exhibits programmed cell death. So this isn't a fluke. This is something we see in many different clades. Well, in Chlamydomonas, programmed cell death can be induced by heat stress. So 42 degrees Celsius is a moderate heat stress for Chlamydomonas. It can also be induced by exposure to toxins, as well as UV exposure. Now, in my lab, we utilize heat stress. And so here is a normal cell And it's a bit out of focus with this. This is like just your basic, light microscope. This is not very fancy microscopy for this image, but you can see that there are two cilia here. And in general, the cell looks oval. It looks smooth. But on the right, after a couple of hours at 42 degrees Celsius, the cilia are gone and the cell is irregularly shaped, and in general, the interior components are breaking apart into bits. I mentioned earlier that programmed cell death is associated with, excuse me, the accumulation of reactive oxygen species. And we can see on the left here, these are rather Chlamydomonas cells, and they're fluorescing green. The chlorophyll is fluorescing in these. And then after heat stress, notice that we have far fewer cells. So several of them have undergone programmed cell death. And then in red is our reactive oxygen species. So wherever you see red, that indicates a cell that is releasing reactive oxygen species, and that's a hallmark of programmed cell death. So there's more cells to die, in other words, okay? Some of these are not gonna make it either. And if we zoom in on that and we merge the green of the chlorophyll and the red of the reactive oxygen species, we get yellow. Because in light, when you take red and green, you get yellow. That's not true of paint, right? But in light, red and green makes yellow. And so when you see yellow here, that means the reactive oxygen species are overlaying on top of where the chlorophyll is found. And so that suggests that the chloroplast may have a role in generating these reactive oxygen species, which is not terribly unexpected. I want to pause for just a moment to say that we know very, very little about programmed cell death in Chlamydomonas. So when I say that it may involve the chloroplast, that's very exciting because nobody's published any of that. So we just don't know very much. In Chlamydomonas, we also see DNA laddering. So when we initiate heat stress, which would be on the left side, and all the lanes going to the right are DNA extracted at later time points after heat stress, you can see that DNA laddering occurs. Okay, so that's very similar to what we see with apoptosis in mammalian cells. And so one of the questions we want to ask is programmed cell death events in diverse organisms phenotypically similar? So I just showed you that in Chlamydomonas, we have DNA laddering, just like we see in mammalian cells. So we want to know more. Are there other commonalities? Perhaps this pathway evolved very early on in the history of life, and therefore there are some commonalities among early evolved organisms like Chlamydomonas and later evolved organisms like mammals. And so again, yeah, our hypothesis is that it evolved early in life. So perhaps it evolved when there was a common ancestor of eukaryotes. But believe it or not, there's actually a lot of evidence for programmed cell death in prokaryotes as well. So prokaryotes exhibit forms of programmed cell death. And so perhaps this evolved even earlier when prokaryotes and eukaryotes had a common ancestor. We just don't know, and we're very interested in asking that question. And so our prediction is that unicellular organisms will have a lot of similarities to phenotypic events of animal cells. And so this project's open right now. I had a student who recently graduated, and so that's an open project. We also, again, want to test that hypothesis about PCD or programmed cell death occurring because of kin selection in unicellular organisms. So our hypothesis, for example, is that the rate of programmed cell death in Chlamydomonas in response to heat stress is going to be related to the genetic diversity of the population. And this was inspired by some studies of the slime mold, which is Dictyostelium discoideum. And I won't go into it for the sake of time here, but there's evidence to show that when these slime mold cells are more genetically similar, they undergo programmed cell death more often. And so our prediction in Chlamydomonas is that a genetically diverse population of Chlamydomonas will have a lower rate of programmed cell death in response to stress compared to a genetically identical population. And so again, if you're not with your kin, you might be less likely to sacrifice yourself, right? If you're around clones and you can detect that those clones are around you, you might be triggered to undergo programmed cell death more easily. And that's what we're trying to test. One major assumption of this, however, is that Chlamydomonas cells can detect their kin. So do they know who's around them? Do they know whether they're in a genetically diverse population or a genetically similar one? And we don't know, but there is some evidence to show that they can detect each other. So here's the experimental design. We have genetically identical population, and we think that the rate of PCD will be higher in the genetically identical population versus that of a genetically diverse population. This is probably one of my favorite projects. And some of you I know will know Armand, who used to work in my lab and who graduated last year. He was right about this time, he was on the cusp of getting some really awesome data. And then you know what happened about this time last year. So he didn't get to finish his project, sadly. He had to go on to medical school. And so this is open and waiting for someone to pick it up again. Although, actually, nobody's been working on it. So it's open. I mentioned DNA laddering earlier. We do see that in Chlamydomonas, and we want to understand how that occurs. And so Chlamydomonas has an enzyme known as endonuclease G, and we think that it might be the enzyme responsible for DNA laddering in Chlamydomonas. And one of my new graduate students is investigating this question. So she is going to characterize the role of endonuclease G and determine whether it is involved with DNA laddering. I mentioned caspases earlier, and caspases are really important in apoptosis. In Chlamydomonas, we don't find caspase genes in the genome, but we do find a related gene type known as metacaspase genes. And there are two metacaspase genes, in fact, in the genome. We have metacaspase 1 and metacaspase 2. And we asked this question previously: will Chlamydomonas programmed cell death be altered in mutants of metacaspase 1 or metacaspase 2? And I'm not showing the results here for sake of time, but we found that when you have a mutant that's only defective in metacaspase 1 or a mutant that's only defective in metacaspase 2, we don't see any difference in programmed cell death. Everything occurs like normal. And so what we think is that perhaps metacaspase 1 and metacaspase 2 are functionally redundant. So one can function in place of the other. And so what might be the answer to investigating this? Let's take them both out. And so if we can generate a double mutant that's defective in both of these genes and then see if there's a phenotypic effect, that might tell us whether these genes are involved in programmed cell death or not. And so I have an undergraduate student named Amber Garcia who is working on this project. And that is the end. I didn't have time to tell you everything that's going on, but I felt like that was enough for one sitting, and I really wanted to save time for questions. So does anyone have any questions that they'd like to ask?
I'm not aware. I'm just wondering because you talked about the role of chloroplasts in the production of reactive oxidative species. I was just wondering, how would you go about isolating them and then analyzing them?
So first of all, I mean, there are different approaches you could take, right? the image I showed you is fairly low magnification, where we showed the overlap of chloroplasts with reactive oxygen species. But I'd like to do some more higher magnification imaging to really see whether those are truly overlapping on a finer scale. I'd also like to take a genetic approach. So there are Chlamydomonas mutants that have defects in chloroplasts. And I'd like to study these mutants and see whether the pattern of reactive oxygen species distribution is the same when chloroplasts are defective. So that would be a genetic approach I'd like to take. I think you're really asking, Tyler, about biochemical approach. So could we extract? Yes, we could extract the chloroplasts. And I am sure that there are enzymatic assays that we could do to determine whether these chloroplasts produce reactive oxygen species. I probably need to partner with Dr. Haynes on that to really figure out an assay that would be workable. But I think it can be done. So I don't know, did that answer your question?
Yes, you hit it right on there.
Okay. Other questions?
I've got another question, but I just want to let anyone else try to tackle it.
Well, go ahead. You can go.
Okay. It's more of like a conceptual question, like related to what you said earlier about like the phosphatidylserines. What is the molecular mechanism for that? Like I just said dendritic, but like how phagocytic cells phagocytize the sorry, I'm stumbling over my words.
That's okay.
Or the cells that are going through that.
So what is the mechanism for the phagocytic cells to recognize the cells? Is that what you're asking?
Yes.
So in general, first of all, we don't have any idea what's going on in Chlamydomonas to allow this. And second, it's a unicellular organism, so there isn't any phagocytosis, right? Mm-hmm. So we would be talking about a multicellular organism like humans, for example, who clearly do phagocytize cells that are dying. So the phosphatidylserine flips to the outside, and there are receptors on these phagocytic cells that recognize that phosphatidylserine. And so that brings the phagocytic cell in contact with the dying cell. And that's kind of one of the early stages of phagocytosis, is you got to make contact first, and then the engulfing can ensue. Does that make sense?
Yes, sir.
Okay.
I was just wondering about like or more about like the transduction of signal and like inside the cell, but it's A-okay if you don't know off the top of your head. I'm sure.
Yeah, I mean, I could tell you all about the pathway. Are you talking about inside the phagocytic cell or, okay, yeah, that I know less about.
Okay.
So good question, but yeah, I'm not exactly sure how that gets initiated. It's a signaling pathway, I'll tell you that, but I don't know the components of the signaling pathway.
Okay.
I'm sure it's in the literature though.
Are the metacaspases relatively conserved across like the other related? I can't remember the genus names or the genus name of your organism.
Chlamydomonas?
Yes. Okay.
Well, so that's an interesting question because Chlamydomonas is a relative of land plants, and land plants are basically those that more or less, right? We're talking about chlorophyta. Okay, chlorophyta are the organisms that have chloroplasts, that have chlorophyll. And basically metacaspases are conserved throughout those organisms. So in other words, metacaspases are very commonly found in plants, in the plant genomes. And green algae are sort of like a primitive plant, in my view. Okay, so they're in the chlorophyta clade. They have chloroplasts. They have a lot in common with land plants in that respect. And that's the reason that Chlamydomonas is a very popular popular model organism for those that are interested in understanding photosynthesis, because they have it in common. Well, if there aren't any other questions,
Does anyone have any questions? Leah, Lauren, Morgan? It's all right. Okay, well, thank you for presenting your research to us, Dr. Gallard, and I'm sure that some of those of us who aren't doing research, or those of us who are ending research should, if they're interested in your projects, we should give you a call, email. Obviously, you can find Dr. Galliard's information on the Sam Houston website if you're interested. So I encourage y'all to do that. There's a lot of research going on. It's always good to have research under your belt. So thank you everyone for coming to this meeting. Thank you, Dr. Galliard, for presenting.
you're welcome.
Hope you'll have a great night, great evening.
Nice to see you all.
All right.
Bye.
Doodles.